Hello, I'm John Gribben from Barts in London. Welcome to the first day of the inaugural international workshop on CAR-T cells. I'm joined by my good friends, Noelle Frey from University of Pennsylvania, my co-chair, David Maloney from the Hutchinson Cancer Center in Seattle. So I think overall, we've all very much enjoyed the day. I think as a workshop, it achieved exactly what we're looking for. That is lots and lots of interactions and lots of kind of audience participation. So we started out the day looking at the two diseases for which there's an approved product. And you, of course, chaired the first session on acute lymphoblastic leukemia. What did you take away from that session today?
So I think it was really an incredible workshop. Everybody, both on the stage giving presentations and in the audience, was very engaged. And one of the things I got out of the ALL session was actually some of the questions that were being proposed by doctors that specialize in other diseases. Yeah. So a lot of the lymphoma doctors were asking questions, trying to correlate it with their experience, and that was really interesting. So one of the take homes that I came with is that with the disease specifically of ALL, we've made tremendous progress. This is now a therapy that offers patients who 5 years ago didn't have good therapeutic options, offers them real hope. But I think it also really highlighted how much further we can go to improve things, to improve side effects, to develop new therapies or combination therapies that can take it to that next level, make it safer, which would mean more patients can participate, and to improve the long-term survival.
Yeah, that's what I took away from thinking about today, the feeling that we're just at the beginning of something really exciting that's going to move on. Now, one of the other things I, of course, took away from hearing you talk today was that your centers had a lot of experience of using different products. And we kept hearing today about differences among the products. And of course, lots of people were in centers where they've only really got experience of using one and therefore don't get that sense of doing. Does the product itself really matter? Or for you, is it often a case of what you have access to at the time a patient comes along?
Yeah, so I think that's a great question. And so I think of the commercial products that we have available and even the experience we've had in products in clinical trials. I think the overall big picture, it doesn't matter too much. I think they're all very effective, but I do think they're different. And that's something that we've learned through the past few years, and understanding those differences in terms of side effects, when side effects happen, how severe they can become potentially, is really important for the clinician at the bedside to understand, and that understanding allows for better monitoring strategies, prevention strategies. So I think, the big picture, I think they're all very similar, but they do have these distinctions that is important to try and figure out.
So, David, in the diffuse large B-cell lymphoma setting, we had a really good long session on that today. There are, of course, two issues. We're seeing the longer-term follow-up on some of the earlier clinical trials, so we're seeing those longer plateaus. And we're also getting more and more of this so-called real-world experience of people using the products that are licensed outside the setting of direct clinical trials. Now, of course, at your center, as in yours, you're doing the clinical products, but you've also got your own academic program of what's going forward. Now, it did come up several times today that there is that kind of ethical issue about how do you decide that you can put a patient onto a clinical trial, particularly in an earlier phase, when there is an approved product? How much does that factor into your decision-making process?
Well, at our center, it's a big deal.
Yeah.
And that's because it's not without repercussions what you do first.
Yeah.
And so that's what I think most people don't understand. And so we're using a limited number of single chain FV CD19 recognition molecules right now. In fact, all the CARs essentially are using the same FMC63 antibody. And what happens is you can clearly get a rejection based on making a T cell response against peptides from that mirroring antibody. So if you really then follow that through, if you get Kymriah or lisocabtagene or iwCAR-T, either of those three products, you're probably not going to respond to the other one.
Yeah.
And so you get one crack at it. So then if you fast forward and say, okay, now I have a completely unvalidated product, never been in humans, it's still using FMC63, and we're at a 1/10 or 100th dose that's going to cause an effect, that may very well, make that patient reject the cells, and then they would not respond. So I have no problem with people being on clinical trials, but I think it's implicit on us as physicians to really present the data to them, saying, look, you might be compromising your ability to be treated. So at our center, personally, if we have something that's phase 1 dose escalation where there's no safety record, there's no efficacy data, we don't think you can go into the population that is eligible to receive a commercial product. So then what do? Well, you take people who couldn't get a commercial product, and there's a variety of ways of doing this. And where this is going to come up is our discussion probably tomorrow when we get to allogeneic off-the-shelf CAR-T.
Sure.
I mean, is it ethical to put a patient on an allogeneic off-the-shelf CAR if you can give them axicabtagene or tisagen or lisocabtagene? And I think it's going to be tricky. So you either have to define a population that is not eligible to get it, like a different histology, until you can get safety and activity, where you're going to phase 3 because you've got a really good product. Then I think you can say, look, this is maybe different. It's maybe now safer. Here's the data that backs it up, the efficacy. Then I think you can do it. So you're either going to have to go into a population that can't wait, and that's hard to define. How do we define those people, that can't wait, or that you could not produce a product? And that's where I think you have to start. And so I actually feel quite strongly about this, much to the irritation of, I think, some other people, just because this is a very contentious issue.
So was there any group of patients that you were hearing today for both approved products, for acute lymphoblastic leukemia, for diffuse large B-cell lymphoma, where you could say, your success with the product that's already licensed is likely to be less good, and therefore you might be a candidate for this clinical trial? Or are you not at the point yet where you feel you can accurately define that?
I don't think we can say that. I think we can say we've got 3 products or 2 products available, one almost, that look, roughly the same. Now you can argue that there's going to be differences in toxicity. You can argue maybe there's differences in efficacy. Some can be used in CNS disease, some can't. So there are some subtle differences. That said, I think that they're more similar than not.
Sure.
What I kind of took away was there's still a lot of room for improvement. Mm-hmm. I mean, even with the 2-year data, we're looking at 40% long-term disease-free survival in large cell lymphoma. So that means 60% of patients were not. So there's a lot of room for improvement. I didn't hear anything today that just said, oh, we've solved it. And PD-L1 antibodies, PD-1, maybe something, but there's clearly a lot of room. And there might be ways we can select patients with earlier on in the course of their disease, less disease burden, that studies from the ZUMA-1 would suggest that they'd have a good outcome.
Sure. Now, David, you kind of kicked the whole thing off this morning with a pretty contentious. I thought maybe you'd lost us all our corporate sponsors in the first 5 minutes, but you were kind of throwing down the gauntlet to the companies in terms of thinking about some of the aspects that make this product quite restricted, in ways in which we could be thinking as a community to how make it more
I think that we're the consumers, right? We're ordering this product. And to some extent, I think the companies should be making it easy for us to do that. And so one of the ways I was suggesting was, disconnecting the production and the selection of getting the T cells from the actual manufacturing process. So I can envision a world in the future where you collect somebody's T cells under some universal product, and then you decide down the road which company you're going to send them to based on whoever has the best product at that time or for your patient. I don't believe we should be tied into somebody's product by the way we collect the cells. Now, that raised a furor of discussion,
I heard that, and
I can understand that.
Well, I hear you, but on the other hand, we all collect cells in our centers under SOPs and under FACT or JC standards. So the standards for which we collect and freeze cells are already pretty standardized, are they not? So is there anything so different?
Well, I think there are some differences. For example, the Juno-Cellular product requires fresh cells because they do a CD4, CD8 selection before they cryopreserve. So that's a difference, right? Yeah. Novartis and Kite both actually can now accept cryopreserved product, at least in Europe. And Novartis they have to, and Novartis was doing it all along. And so I think Novartis is the closest to being actually have something that is close to what I'm already proposing. But I think the other the other manufacturers are going to need to take notice at some point, because there is going to be competition, and people are not going to want to be stuck with especially if you're collecting prophylactically or collecting people beforehand, would be then stuck with that manufacturer if it turns out someone else has a better idea. So I can envision the future will be some universal collection, either by a third party or by our own centers. There's a lot of logistic issues, right? How do we store the cells? Who pays for it? Do we write that off? I mean, but you guys are already doing that in pediatric ALL, is my understanding.
Yeah, so I'm on your side on this issue for sure. I think being able to collect a product and use that product for manufacturing across different opportunities would be tremendous. And I think while that does pose a tremendous challenge to the products that are already approved, I think it's a nice thought to have in the back of our minds as the field moves forward. And not only does that make things logistically easier for the center and the physicians, but I think it would really impact outcomes for patients too. I mean, you have a patient who you meet at the time where they actually need this therapy. That means they have relapsed and refractory disease. Their T cells are probably exhausted. They've been through a lot of therapy. So if we can have a product, develop a product from a collection that happened at an earlier time in that patient's course, that patient might actually get to treatment, whereas otherwise they may have not. And the quality of the product is likely to be improved.
But I think you also pointed out some really key things. I think we shouldn't have 4 or 5 REMS programs with CAR T cells, right? We shouldn't have multiple criteria for how we assess toxicity. And that came out that was pretty contentious as well, right? So I think everyone is kind of agreeing we need to get on the same page. And probably with therapy as well, right? How do you intervene? Who should you intervene in? The products are clearly different, but there's more similarities than not in terms of, I think, where you intervene.